also developer these pests, or evolving to try to promote?
I think there's a vision, but I'm not sure if that's something which we can make reality. But especially if you go to the farmland and everything, we use still a lot of pesticides. We need them, but what's the aim to get that lower? They get more expensive, and they're very bad for the environment. So how can we find ways to lower the pests? And I'm not saying this is the best way to do so, but I think we need to figure out all ways we have to do that. And this one does work quite tempting for farmers to do so. That's why I try to do that. But I will need a lot of scientists to do that. I'm still just a tech guy in a biology field. So
brilliant. Thank you very much.
welcome.
Okay, next up we have Vicky. Hi, I'm going to be presenting on a very generously named identification machine that we've developed at Forest Research. So to set a little bit of context behind this, we have a problem in the UK with the 8-tooth spruce bark beetle. So this is a native beetle for Eastern Europe and Asia, and it's a major pest of spruce trees. It's usually a secondary pest, so it feeds on dead and dying spruce, but it can build up into really, really high numbers, and has actually decimated forests across Europe through these things called mass attacks, where the beetles will repeatedly attack healthy trees and weaken those defences until they eventually kill the trees. This is an example of an event in the Czech Republic on the left, and just to get a sense of scale, this is a big pile of beetles from traps across the Artvin forest in Turkey. So that's about 100 million Ipsipographus, all in one site, all essentially on at the same time. So we found a breeding population of Ipsipographus in 2018, which is obviously super scary, because we have about a £3 billion forestry and wood processing industry in the UK. And the the softwood portion of that is predominantly spruce as well. So this is a spruce map of the UK, and you can sort of see that the spruce is concentrated in Scotland, Wales and the north of England, and this incursion was found in the southeast of England over here, and further research showed that actually this wasn't an interception from a port, but these beetles were actually being transmitted carried by the wind from continental Europe. So what are we doing about this in Forest Research and in the Forestry Commission? These beetles are attracted to aggregation pheromones. So we have cross-vein traps, as you can see in the top left there. And in 2019, we set up a monitoring network around that first incursion site. And that has since expanded as a monitoring network across the UK into Scotland and Wales this year. For every single one of those traps, the beetles are collected and killed in glycol, and they are sent to us in Forest Research in Alice Holt, where a team of entomologists identify those beetles and count how many beetles are in each trap. This usually, you can see by the large portion of whites, this is usually we don't get any beetles, but in the southeast of England, occasionally we get breeding populations. So far, we've been able to eradicate every breeding population we found, but we trap on those sites where we find breeding populations for 3 years. And those are where we get sort of quite high, relatively high for us, numbers of beetles. Can look something like this on the left, where we get up to about 8,000 beetles within a collection event. And we've got a team of entomologists right now doing manual clicking to count how many beetles they have in each of these traps for each collection event. It's really reliable. So like when we've actually tested between our counters, they've got really consistent results, which is really nice, but it is really time consuming. So we've got a gold standard of these manual counts, but it's very resource intensive, it's very slow. So it can be anywhere between 3 and 6 weeks between a beetle flying into a trap and actually reporting on that beetle being present. So can we automate this process? That is where the automated insect identification machine, or AIIM, comes in. So we have a CNC machine set up in our labs, which essentially is a camera fixed onto an arm that takes images at fixed intervals. And then there's a bespoke FR pipeline to stitch those images together. So I'll just talk you through the pipeline for the model. So yeah, we have this camera on an arm, and it stitches high resolution images together. So you get a sample that looks something like this. And you can split your sample into multiple images and then combine the results at the end. Important to note here is that these samples, regardless of how much we like spread them out, it's still fairly dense. There are overlapping specimens, and it's a multi-species mix within this sample. So what we've been doing is training a deep learning model. So we've been doing polygon masks of whole beetles, including partially obscure presentations and where there's overlaps. We have, because this is a pheromone law in there isn't a ton of bycatch. So we've gone for these very broad categories, which we can discuss later, so that we've got enough of a sample size within them to train the model. And we're using a mask RCNN architecture from Detectron 2. We've got standard augmentations in there. So that's like random rotations, using a little bit of random scaling, flipping images, adding a little bit of brightness and contrast differentiation. And then we also have instance level copy paste augmentations that we've developed for this, where you take an IPS from one image and then paste it into another image. It's got a random chance of pasting 1 to 3 IPS 50% of the time. So we also have rare classes upweighted in this model. So what have we actually done so far? We've got currently about 7,500 total annotations, which I recognise for this room is quite a low number, but considering how long it takes to do that per pixel segmentation, is actually really good. And we take these large images and split them into 4 rows and columns. So you take those original 21 images and split them into 336. Those overlap so that we can stitch them back together at the end. But it's better, we found that the model is better at detecting smaller things if the image is split than across a large one. So what's the accuracy looking like? The overall quality of the detections across all the confidence levels are presented here. The best categories for this are the debris, the IPS, and the other beetles, but we're still building that training set and going to do a retrain as well. What we've noticed from like manually inspecting the detections is that this is missing several small and medium individuals. It's really good for like large beetles, really good for like large other insects, but it's missing the little tiny ones. Luckily for our study species IPS, they don't vary hugely in size or shape, so they're very forgiving in their presentation. So we're not as worried about missing those small ones for the purposes of this. If you look at a row normalized mass, each row on this diagram represents of the times that the image was correctly segmented, what did the model predict that object to be? And the diagonals on this are the correct predictions. So it's doing really well when it's segmented debris, when it's segmented IPS correctly. It's doing really well for debris. It's doing pretty good for IPS considering the size of the training data set. And it's doing quite badly for like that miscellaneous category and other insects, which also sort of makes sense considering how many things we're lumping into that category. So comparing it with actual manual counts, which is that gold standard I mentioned earlier, this is just looking at 11 samples across the spread of sample sizes that we normally get in. And the difference between the manual counts and the predicted number of beetles is actually like relatively small. We're quite chuffed with that. It's pleasantly surprised, I think. And there was no significant difference between those counts. So the whole point of this was to try and save us some more time so that we could scale things up and get entomologists doing entomology and not just counting. And AIIM is significantly faster than doing those manual counts across the board, which is, again, really reassuring and encouraging. So can this be automated? It looks like yes, which is very nice. So in terms of upcoming work, this project was originally developed in parallel with a smart trap in the field and a lab-based protocol. We've split that. So what I've presented today is the lab-based protocol, because the smart trap wasn't as effective as we were hoping it would be. It's quite large. We had like several problems with it, which I can talk about later if that's helpful. So what I'm partly here for today, predominantly here for, is to ask you if anyone would like to collaborate on a future project that combines these slot traps that we have at ports with our learning from the smart trap that we've developed, and creates a new smart trap to go into ports that's adaptable and fits onto the slot traps that we already use. The next steps for me are to develop a user interface for the lab protocol. It's currently all up on Kaggle, which looks a bit scary for people who don't code. Yeah, the requirements of the smart trap would be to capture the beetles, contain the beetles, kill the beetles, then image them and securely transmit those images to us at Forest Research, because these are sensitive data. So if anybody would like to chat with me afterwards, this is my email address, and the QR code will also take me to my email address, so please do. And I want to thank everyone involved in this project, particularly Barley Rose and Ruth, who actually set this up, and I have inherited this from them. So yeah, thank you.
Fascinating, thank you. Does anyone want to kick off the questions? Yeah.
Maybe I missed information. You said Barney would still make it to bring even the spruce tree make it too.
Yeah, but you're correct. So spruce was planted in the 50s and 60s across the whole of the UK, but mostly in Scotland, Wales, and north of England, because the south of England doesn't have very good soil type for it. Not that really stops people, but it's not the best place for those to grow. But they were planted as a crop, as a timber product in the 50s and 60s, and are reaching maturity now. Yeah.
Yeah, but the product is very contaminated. So I like the impression to stop the market because of something that is coming like a natural fragrance.
So I totally understand, but the spruce industry for us is a massive economic resource. It's actually really important for like our national security as well, to be able to produce our own timber and like be self-sufficient in that, and also be able to like export and import. So the reason why it's important for us to be preventing this species from spreading is because our timber is so important to us economically and in terms of resilience. Thank you.
Yeah, Leah?
Against financial it also has to be a essential population
Yeah, absolutely. Thank you.
Yeah, Frederick.
You plan to expand what we did on the best mark vehicles.
Yeah, so part of the very broad brief for the port traps is also to look if we can develop a pipeline that also has a rapid response to any quarantine organism, which is a very, very broad brief for that. But that is the hope, is to take this and prepare like readiness plans for if there are future pests that come in, now that we know that pests can be wind dispersed across the channel.
Do you want to have the last one?
Yeah, so all of